Then finally the results same. How much amount of the respective DNA like fragments has been deleted or inserted. This is the software. We need to check the respective sequencing results data. Anyone have with the respective sequence means you can control, you can analyze with the
anyone have YouTube channel
analyzer.
yeah
This is freely available too. First, initially we get in the form of chromatograms. In the respective chromatograms, we need to paste here, and then finally we need to calculate the percentage of single mutation. Okay, this is the final sequencing results. After that, we need to go for the respective trait analysis. Okay, first we have designed the guide RNAs. First, we need to fix the target, then design the guide RNAs, then followed by the guide RNAs, and clone into the vector, respective vector. Then, in the case of animals, several techniques, microinjection, then transfection, electroporation techniques are available. In the case of plants, particle bombardment. But in India, particle bombardment technology is a failure things. Agrobacterium mediated transformation is a well-established technology, but instead of agrobacterium technology, we can prefer electroporation also. But electroporation, only the single cells. We need to isolate the particular cells from the leaves. For the best example, protoplasm. Or the protoplast cytometry, we need to isolate the protoplast, then we need to transfer to the respective constructs. Then finally, we need to emerge the respective plants or the respective species. After that, we need to directly go with the PCR analysis to check whether addition is there or not. To compare with the respective primers, first thing. Then second thing, then sequencing. The sequencing is the results. We need to compare with the respective primers, the respective base addition or deletion. Indel mutations, we need to check with the indel mutations. Then we go with the respective trait analysis. In the case of plants means we need to focus with the respective quality. In the case of animals means we need to check with the respective trait. In the case of, I will show the next. Validation. After that, completion of addition experiment, we need to check the validation. For the case of animals, we need to go with the Western blot analysis for the complete validation of the respective proteins. Western blot analysis. Or otherwise, we need to go with the whole transcriptome analysis using RNA sequencing analysis. In the case of RNA sequencing analysis, all the documents will be provided by the company itself. Then the company itself, they can provide the reference sequence. They will itself, we need to spend some more amount. Company people itself, they can give the respective results in the form of tabular column or the form of graphs. In which pathway is an upregulated or downregulated, they will itself analyze. We need to isolate the RNA from the respective sample, whatever it may be, the microbe, fungi, micro plant animal. Then go to the sequencing analysis, RNA sequence for the whole transcriptome. Otherwise, we need to check with the RT-PCR analysis whether the particular gene has to be upregulated or the downregulated. We need to check with the respective pathways. For the best example, in the case of plants, we need to check with the respective secondary metabolite pathway. Correct. Otherwise, we need to check with the respective apoptosis pathway, or in the case of some other pathways, cell cycle regulators like that. We need to check with the respective regulation. This is for the RNA sequencing and then RT-PCR analysis. Then followed by the protein analysis. In the first thing, protein analysis, we need to check with the protein analysis using to isolate the protein. Then go with the STS page followed by the Western blot analysis. This is the functional validation where the respective protein upregulated or the downregulated actually takes place. The tissues are the species. Correct. Then this is software. This is a bioinformatic tool, the string database is a software. We need to check with the respective proteins, with the how the protein were combined with the some other protein groups. This is the interaction between the some other groups, how the protein were interacted with the some other respective proteins. This is an original software, then bioinformatic tool prediction. We'll see the upcoming slides. Then, in the case of plants, we need to check the physiological parameters. In the case of recently developed nowadays plants are always plants are green in color, correct? Why? Chlorophyll. Chlorophyll is there. Oh no. Chlorophyll molecule which absorbs the blue-green light, then emits some kind of green signals. That's why plants are green in color, correct? But nowadays, the best thing is a visible marker in the case of plants, not in animals. We need to check whether the particular experiment is working or not. We need to check the reduction of the chlorophyll content. Then we need to generate the nutrients. To check with the reduction of chlorophyll pigments from the plants. This is the best method to check the physiological parameters. After that, the plants will be pale white in color. The control is in green in color. The editor should be the pale white. We need to check with some kind of parameters. But we need to check with the physiological parameters, like we need to check the entire morphological traits. We need to analyze the shoot and the root. In the case of, we need to reduce the auxin regulated pathway, means in the case of tryptophan is a major amino acid for the production of auxin. In some kind of monoxygenase family, some transcription factors are there. YUCCA is a positive regulators for the upregulating the pathways to enhance the production of auxin in the shoots. Because auxin were produced only the shoot apical meristem, then followed by the transportation. Auxin can transported towards the entire plants, mainly nodal segments and shoot root apical meristem. This is the physiological mechanisms where the auxin play a major role. But in transport mechanism, two transporters are there: polar transport and then non-polar transport. Influx and then efflux carrier molecules are there in the plant itself. Influx means like auxin outside to the inside will transport towards the inside to the cell. Efflux means outside to the cell. In outside, pink prenoid proteins are the major protein to downregulate the auxin content. Okay, is it non-polar transporter molecule? Pink, PIN. Prenoid is a protein regulatory complex. This is the protein where the negative signals for the auxin signaling and then transportation mechanism. We need to downregulate the respective twin protein from the cytoplasmic membrane. We have did the experiment, we got the results, but we need to check the physiological parameter in the aspect of plants, means we need to quantify the respective auxin content present in the shoot followed by the root. Then only we can compare with the how much amount of auxin were synthesized from the shoot and then transported to the root. We need to compare with the results. Then only we can conclude that they have precisely edited the particular pin protein. Then it should be upregulated by the auxin regulatory transport mechanism. We need to check the physiological and then biochemical parameters. Okay, after that, in the case of animal means we need to grow the respective animal in the laboratory conditions or in the animal house conditions. Then we need to check with the respective whether the particular disease has been cured or not. Correct. Similarly, we need to grow the respective plants from the seedlings to the hardening stage. Then we will conclude the conditions. We need to quantify the respective parameters. The complete results will occur in the end of the fruiting stage. This is a physiological and biochemical agronomical traits. We need to morphological analysis and then physiological analysis and then complete the results with the another parameter. Okay. This is the string database. Like we need to check the here we have some tricks. We have already edited some lines. Correct. Already edited some lines. We got chromatogram. We got the ABI form along with the PDF form. ABI form is an editable version. We go for the bioedit tools and then we can retrieve the respective sequence from the software itself. After that sequence, we need to create the homology modeling of the respective 3D structure of the protein by using Swiss homology modeler. Okay. We need to generate the respective protein structure. Then finally, we can paste into the software. Then this software will analyze this mutant protein, how this mutant protein will interact with some other group of proteins. Okay, we need to generate first mutant protein 3D structure analysis. Then we need to incorporate using this software. Then finally, we'll get the results, the form like this. Like in between, the DREB, the dark red color protein is my protein. Just for the example, how this protein were interacted with the some other proteins like actin. Actin is a normal control normal housekeeping gene, you know. And then how this protein were interacted with the various soybean related proteins, we can easily identify this using this software. But we need to generate the 3D model. Okay, we need to generate the mutant version of the protein by using Swiss homology modeler. We can get the nucleotide sequence from the ABI format and then paste into PDB sum. And then we need to generate the Ramachandran plots whether the particular design protein shows the more than 95% of the similarity of the respective proteins. If okay, we'll go with the respective string analysis. Then how this mutant protein will interact with the some other original proteins. We need to check with the Ramachandran plot analysis because this is also a protein, but this is a mutant version. In mutant version means in some case some bases will replace or some bases will added, deleted, inversion, whatever it may be. The mutant protein is there, normal protein. We need to check with the alpha helix and the beta helix. We need to quantify it using Ramachandran plot analysis. Correct. Basically, we need to analyze the protein by using Ramachandran plot analysis only. Then we go for the string analysis. After string, our experiment is done. Then we will go for the what? Manuscript writing. Correct? Once our product is okay, then we go for validation of our respective product by using market. Correct? In the case of research, all the data were collected from the and then we'll show it to our respective PI. If PI is okay, we can publish or otherwise keep quiet. Okay, that's it. But we need to, this is a two-step process. We need to generate the mutant version of the protein structure. Then we need to incorporate into this software. Then we need to generate like this database. Okay.
యూట్యూబ్ అని సార్ మీరే వస్తారు
SnapGene is a freely available tool for 1 month, one
అన్ని
accession.
తీసుకో
But we need to pay some amount for an annual subscription base. We need to get the good full accessible of the SnapGene. But in 1 month basis, we can view and then we can edit the particular gene. Then just we make some kind of analysis on it. In the case of subscription, all the facilities, all the parameters will appear in the payment basis. SnapGene is a normal cloning. Yesterday I showed some image, how the vector has been digested, inserted by the aspect of graphical representation, in which basis has to be deleted, inserted, it should be given by the respective software called SnapGene. Okay. Mainly cloning and then PCR amplification products will appear in this software. Just go through the SnapGene version and then easily access your respective registered email for 1 month basis. You can alter your respective email ID if you want. Okay. Yes. I don't know about when I was working previously, I don't know about this software. Previously we were working in SnapGene version, but this is a fully non-profitable organization software. Is an intron exam graphic maker. When I was working in GKVK at the time only, I could know about this software. You can easily make the linear diagram. Like I'll show the home page. Like this. Chapter 1, the graphical representation of the respective gene sequence. How the down arrow, the color should be the intronic and then exonic sequence. Sorry, exonic sequence. The down arrow, like joining sequences are the intronic sequence. But in the graphical abstracts, we are making, in the case of publications, they were asking about the graphical abstract submission. Graphical abstract is a major important thing for the publication aspect. At the time, we need to make the good graphical abstracts. For that illustration, we need to use this kind of softwares to make the linear map of the respective targets in the edition side. This is a software. Then see, like this, the results appear like the gene name, intron, exon, the position of the sequence will appear like this. Okay. For this, one thing, for this, we need to identify the 5' UTR intronic sequence, exonic sequence from the chart. Sorry, yesterday we just phytosome. We need to copy and then paste into the respective site, and then we need to generate the graphical abstracts, graphical illustrations, like before I showed that. Like we need to copy, like parallelly we can use access the phytosome also.
ఎందుకు Okay.